So hello again, welcome everyone. Thank you for joining us for the Sight and Sound Bites webinar today, this Friday. My name is Carrie Fogel. I'm the Senior Director of Development for Foundation Relations at the Ioneer Foundation. We put on these webinars every month, as many of you know, to feature our amazing faculty and experts talking about different conditions and topics related to hearing loss, like today, vision loss, head and neck cancer, sinus, allergy, balance disorders, and conditions of the voice. So today's presentation will feature a discussion about restoring hearing and regenerating hair cells. Our speakers today are Dr. Peter Santamaria and Dr. Melissa McGovern. As a reminder, we don't use the chat function as part of our webinar, but we do ask that you submit questions to the Q&A box at the bottom of your screen. You can submit them at any time, and I will hold all the questions until the end and make sure that we get them answered to our experts during the Q&A portion of the talk. If we somehow don't have time to get all of the questions, or if your question is more personal in nature, then we will send those questions over to our experts following the program and we'll get an answer sent to you directly. And as always, there will be a recording available of this webinar on our website. So if you want to refer back to it or send it to anybody that you know might be interested, it should be on our YouTube channel and sent out via email later this weekend or early next week. So before we get started, I must introduce our two speakers, as they're both, new faculty members to us in the department. We're really excited to have them both here talking about different topics related to hearing restoration. So I'll start with Dr. Peter Santamaria. Dr. Santamaria is a surgeon scientist subspecializing in disorders affecting hearing, balance, and the facial nerve. Along with his role as director of Spark Global, he co-leads the CREATES Lab here in Pittsburgh and is chair emeritus of the American Academy of Otolaryngology's Medical Device and Drug Committee. He recently joined the team at UPMC as division chief of Otology and Neurotology and became the vice chair of clinical and translational research, and comes to us from Stanford University and just started this past summer. And Dr. McGovern, Melissa McGovern, is our co-speaker today, and she's a molecular biologist who investigates hearing restoration following the loss of sensory cells in the ear. She received her bachelor's degree in psychology from Illinois College in 2013 and her Ph.D. in pharmacology and neuroscience from Southern Illinois University. Dr. McGovern completed her postdoc at Baylor College of Medicine and then joined our faculty here last summer in May 2023. And her lab investigates the genetic barriers restricting the hair cell regeneration in the mammalian inner ear. So really cool stuff. Looking forward to a great talk. I'm going to turn it over to Dr. Santamaria to get started. Thank you.
Thank you, Carrie. And it's really great to be here and to have so many people participating. And I look forward to giving everyone an insight about how, of course, important hearing and hearing research is. And it's kind of the foundation. And one of the things that attracted me to come here was how great everything is here. So let me tell you a little bit about why I think it's important. So let me just work through this slide here. So as was kind of alluded, and you could probably tell as soon as I opened my mouth, that I did all of my training in Australia. I was born, raised, and trained in Western Australia, and did fellowship, and then moved to Stanford for further fellowship training and became faculty there. We're a part of quite a large clinical team here, and we're growing. We're openly recruiting, so we're going to grow further. But we have excellent clinical leadership from Dr. Bazura, Dr. Perez, and also Dr. Furman from Otolaryngology that handles a lot of balance disorders, and Dr. Palmer on the audiology side, as well as Megan and Maura, both provide a lot of medical otology services in our department. So we really are a big and growing team. I'd like to start with this quote from Helen Keller, and she's kind of famous for this, saying that blindness separates people from things and deafness separates people from people. And this kind of makes it, kind of hits home to me about why it's important for what we do. Obviously, no one wants to be blind, but of course, that separates you from the world and the things around you. But no one knows that you're deaf or you're hard of hearing. It's a very invisible disability. And what we notice with people with hearing loss is you change who you are as a person. And over time, you often become less social, you tend to withdraw, and you tend to have, you change in your family relationships, your marriage, and all these things happen very gradually. So it's really something that influences our ability to connect with people and to communicate. And so, we don't want to see that, and that's why we do what we do. And it really is common. It affects 1 in 3 over the age of 65. It's the second most common presentation in people over 65, and it's the third most prevalent health condition. Everyone has hearing loss or knows someone with hearing loss. And if we look at the cost that economically around the world, there's $750 billion in attributable healthcare costs to hearing loss, and that's a big deal. One of the big reasons why it really affects us as we grow older is there's a direct link between hearing loss and falling. If you're not hearing and aware of your surroundings as much, and particularly if your balance is affected as often goes along with your hearing, you can have very impactful events in your life that particularly with hip fractures in the elderly, there's a very high mortality rate associated with that. So hearing has an impact that goes larger than hearing. One of the things that scares me and scares a lot of people is its association with dementia. Now, this research was published in 2017, so it's getting a little old now. So hopefully we're all aware of it. But there is many things in life that are attributed to dementia or cognitive function as we grow older. Earlier in life, it's really there's a factor around how much education you get, and later in life, there's things like smoking, depression, being inactive, and diabetes. We all know about these things, but are we aware of the impact that hearing loss has on our cognitive function? And if you look at their 9%, it is regarded as the largest reversible variable or cause that could lead to cognitive impairment as we grow older. So we need to take care of our hearing. But why does hearing loss impact cognitive function? It's because the simple way to describe it is your brain needs to work a lot harder when you can't hear. And so the cognitive load that's going on into your brain is much more difficult if you're trying to interpret a very different sound that's coming in than you're used to. All these things can happen, and there's research to show that your brain structure can change, that you're reducing your social engagement that I alluded to before. All these things lead to more cognitive load that can then lead to impairments in cognitive functioning. So I want to emphasize that hearing loss is a modifiable risk factor that we can use to prevent cognitive decline and dementia as we all grow older. So there's a lot of barriers to accessing innovations in hearing loss. So of course, there's an awareness and understanding gap. There's some issues with, especially in the US, at accessing some of these technologies, even hearing aids. And also there's some gaps in technology that we're looking to innovate around. And one of the biggest things that we're faced is that, if I wear glasses, which are eye aids, I'm considered smart. But if you wear hearing aids, the old stigma was that you're growing old. And so, even, presidential elections are at the forefront of us right now. But, this was a big thing when, I guess, Reagan was going up for election, that, he was seen to be wearing hearing aids. And there was speculation at the time whether or not he was cognitive declining and whether or not that would be an issue for his re-election. And that still exists today. I'm happy to say that stigma is decreasing. So as Carrie mentioned before, I moved from the Bay Area, and I would find that most of the young people there were very excited to be wearing hearing aids. We were in a technology area, and everyone was wearing some sort of earbuds or a wearable. And so adding a hearing aid was not a big deal. But I realized that still is a barrier that we need to break through. And like I said, it's now getting cool to wear something around your ear. You know, we're going away even from small earbuds to having really big and visual headphones and really looking at sleek styles. And most of these can be converted into hearing aids. So, you know, we just need to think a little bit differently about hearing loss. It's not a disability anymore, and it's a way that we can connect to the world. And so, you know, it's a long while since we've evolved from this ear trumpet that was first used, and even the middle one, which is our stock standard hearing aid today, where even the difference between the second and third picture, you can see some very sleek changes in the design that even as a hearing aid doctor, I don't even notice people wearing hearing aids anymore, with the very sleek standard designs that are going around today. And they come in a lot of different shapes and sizes. So they can be all the way on the left, they can be completely hidden inside the canal, and invisible, or they can be just placed in the canal, or they, you know, the standard is to have an open fitting where they're fitted behind the ear with a clear colorless tube connected to the earpiece sitting inside. And really they're getting they're getting less and less visible. So the other innovation that's happening is making hearing aids even more invisible. So you can see up there as an example, the lyric hearing aids hidden completely inside the canal. There's another version called the ear lens, which is a rather from acoustic, which is sound amplification, using hearing aids driven by light or magnetic to drive the drum to get an even better high frequency response. So there's a lot of innovation that is still happening in and around the hearing aid itself. This is an article that we published relatively recently, going through all the innovations related in hearing aids or hearing augmentation that could be attributable or used in the age-related hearing loss setting. And they really go from not just the hearing aids themselves, but the way they interact with the environment, and implantable devices, which is something that I'll just very briefly touch in here, but of course happy to provide more information later on. And so, as you can see there, we're going into an innovative area where things are becoming much, much cheaper and available even online, which I'll touch on. I think one of the biggest innovations is going to be hearing aid pricing. And so it's embarrassing to say that for a cost of eye aids or glasses, you may pay around $100 or $200. But for the hearing aid technology, sometimes you can be out of pocket in the thousands. And that really doesn't match what the payments of seniors are getting through Social Security and retirement. It really is a big financial impact. And if you really want to look at a really neat comparison, which is what one of my mentors used to tell me a lot, is that really the markup on hearing aids is really equivalent to the markup on movie popcorn. It's really nothing that's really sold at 20 times the cost these days. And so we are really in a field that's ripe for disruption, which is really exciting. You know, in 2017, there became this legislation that changed the way that we describe hearing aids to make them available over the counter. And now we're calling these things PSAPs, or personal sound amplification devices. And these are really impacting the price of hearing amplification. So you can go onto Amazon or any other online store, and you can get a hearing aid. They may not be called a hearing aid, but they're called sound amplification devices, or PSAP. And a lot of these are self-fitting, and you could spend as little as $50. Some of the more custom fit ones that you can also get through Best Buy and other major chains can get into the hundreds. But really, there is something that hopefully will meet most people's budgets. But also, you know, the important thing is that we just need to be really aware that hearing aids don't need to be in the thousands, or hearing rehabilitation doesn't need to cost thousands. And you can see already in the last 5 years how dramatically the prices are changing and reducing. I think the other exciting innovation that's happening is the convergence between hearing aids and this term hearables, or a wearable hearing aid. And so, you know, the hearing aid can do much more than just provide sound. It can measure our heart rate, it can measure our body temperature, it can count our steps. And I think as we move into a wearable or earable generation, we are going to see many more innovations that are going to make the hearing aid more accessible to the average person. We know people that are even working on the ability to translate. So if you go overseas and someone's talking to you in another language, that this device could potentially translate the sound into your ear. And so if you look at the world of where that's going, it really is exciting in how it opens it up. There's the connection between hearing aids and your phone, your personal device, and them becoming smarter. These are some of the things that are out there now. They can be an intelligent personal assistant to remind you where you need to be, your calendar. As we meet people, it can remind us who these people are when they see them. And as I mentioned, language translation, performance tracking, fitness, health monitoring, all those things can be connected. There's research that I was involved in the Bay Area where even within one of the hearing aids is a fall detection device, so that with elderly people, obviously I've just mentioned the link between falling, so that if you were to fall whilst you're wearing a hearing aid, it potentially sends a signal to your loved one or your care team to come and help you out. So we really need to think about hearing aids differently from that old ear trumpet. And one of my favorite things to talk about and to show a fellow Australian, this is Graham Clark, who is one of the people credited for inventing the cochlear implant. And the cochlear implant is a device that we place during surgery, and has a little wire electrode that goes around into your inner ear and converts acoustic sound into electrical sound. So it's really a great device for people when the inner ear is not functioning and we need to bypass that and electrically stimulate the nerve ganglion. And that, as I said, you can see that from the year, was invented in the late 70s. And so we have decades and decades of research there. So it really is not a new device anymore. It's an old device. And when we're talking to patients about cochlear implants, we're really doing that with decades of experience. And that's just showing you the picture going there. You can see on the left there of how a cochlear implant is typically placed. And it can go in and inserts an electrode wire into the to the spiral of the inner ear. And really, that's probably an older picture now, but they really just look like a big hearing aid. And so really, these have changed the way that we manage hearing loss for people that can't wear hearing aids. So it gives me great pleasure to really segue into what Dr. McGovern is going to talk to you about. And the excitement around Pittsburgh is, we have a truly comprehensive group that works on hearing research. We have a lot of people, and I hate to call it basic research because it's more discovery research. It's certainly not simple. And then we have a really excellent clinical team, and we have the people in between that are trying to work out how we can get these discoveries that we're making into the clinic and provide that back in. We have lots of connections into developing countries. You can see down the corner there. Our work together in communities in Mozambique. We have community work that's going out led by Dr. Palmer further out into Western Pennsylvania, and all that centered around people like Dr. McGovern, who are incredibly well funded and talented to solve these problems in hearing loss. So, on that note, I wanted to hand over to Dr. McGovern.
Hello. Thank you so much for the introduction, Peter. It's a great overview of kind of what's currently going on and what the current clinical options are for hearing restoration. And so now I'm here to talk to you about some of the basic science that's going on here at the University of Pittsburgh, where we're really trying to figure out how we can regenerate the sensory cells in the ear called hair cells. And so these cells are inside an organ called the cochlea. The cochlea is part of the inner ear, and we can see this pointed out here with this bright red arrow. And it's a coil-shaped structure that is part of the inner ear. It detects sound. And if we take a cross-section through this, we can take a look at the different areas inside the cochlea. And so typically, the mouse ear is broken down into 3 terns: the apex, middle, and basal term. And so these aren't distinct independent regions necessarily, as much as you can think about it kind of like a spring, where you have one end and it slowly coils through down to the other end. And so that's kind of how the cochlea works as well. However, if we take a closer look at one of these areas of the ear, we can see that there's several different components inside the cochlea, and the area of the cochlea that my lab is very interested in is called the organ of Corti. The organ of Corti contains these sensory cells called hair cells, and these are the cells that actually detect sound and tell the brain about it. These hair cells are surrounded by cochlear supporting cells, and these supporting cells, as you know, their name indicates, they really kind of provide support and function to the hair cells. Unfortunately, the loss of these sensory hair cells causes hearing loss. And so the idea of a potential future gene therapy is to figure out how we can regenerate these sensory cells, these hair cells, from the supporting cells or other cells in their environment. And so we know from the literature that the immature ear in the neonatal mouse can actually spontaneously regenerate these hair cells. Now, this time frame corresponds approximately to the end of the first trimester in human development. So this is very, very early, and the organ can't actually detect sound at this time. So it's very immature still. We can take a look at this by labeling the supporting cells here in red. We can permanently label them with a dye, and then we can take a look at what happens when we kill the hair cells, which are labeled here in green. Again, we have this one row of inner hair cells and 3 rows of outer hair cells, and if we kill the endogenous hair cells, we can ask whether or not the supporting cells or the adjacent cells are turning on hair cell proteins, which we can detect with a marker shown here in green. When we do this in the neonatal cochlea, or the immature cochlea, we can see that there are co-labeled cells indicated with these white arrows here. And so these cells started out their life as a supporting cell. We expressed that dye in those supporting cells, and now they have activated hair cell proteins that we can detect with this green marker. We can quantify the number of these cells and see that if you kill the original hair cells, these supporting cells can spontaneously regenerate the hair cells. And so we have an increase, a significant increase in the number of these co-labeled cells. Now, unfortunately, the mature ear in all mammalian species we've ever looked at, including mice, humans, guinea pigs, etc., they do not naturally regenerate. So here we can visualize the hair cells through their stereocilia bundles. These are these little white V-shaped structures. They look like little hairs that come off the top of the hair cell and actually are where the hair cell gets their name. And so we can see that there are 3 rows of the outer hair cells and a single row of inner hair cells. Following various damages, including loud noises, certain medications can cause the death of these hair cells, and the natural aging process can also cause the death of these hair cells. When these hair cells are lost and we accumulate more and more of these hair cells being lost, this leads to hearing loss, as Peter talked about just previously. And so these don't naturally regenerate following damage, and so this is a permanent hearing loss that we experience throughout our life. So because these cells don't naturally regenerate, this leads us into a discussion of genetic reprogramming. And so this is kind of the basic underpinnings of how we can potentially develop a gene therapy. So to kind of explain what genetic reprogramming involves, is we can think of the various cells in the ear, the supporting cells and non-sensory cells, we see them here in green, and the hair cells are in blue. Now, in all organisms, so all of the cells in my body, all of the cells in y'all's body, all of the cells in a mouse, we all contain the same set of genes in our DNA in every cell. However, different cell types only express a certain group of genes that make them that cell type. So essentially, a supporting cell expresses certain genes, and that makes that cell a supporting cell. A hair cell expresses a different set of genes, and that makes that cell a hair cell. So when we're talking about genetic reprogramming, the question is, how can we activate hair cell identity in a non-hair cell, such as a supporting cell, sufficiently to be able to convert it into a hair cell identity? And so the way that a lot of gene expression is controlled is through things called transcription factors. A transcription factor some people may have heard of is 801. And transcription factors control cellular identity by binding to the DNA. They do this at places called enhancers and promoters, and this drives the expression of something called a protein coding sequence, and that produces proteins in that cell. And so if we have hair cell proteins expressed, it's going to be a hair cell-like cell. Now, 801 is expressed in developing hair cells, and we know in the immature cochlea, similar to that spontaneous regeneration we talked about, that if you express this one transcription factor, you can get conversion of non-hair cells into hair cells. And again, this is a very early immature ear. And so we see here that we have one row of inner hair cells and 3 rows of outer hair cells, and we're detecting 2 different hair cell proteins. If we express 801, that transcription factor, in the non-hair cells in the immature ear, we can actually get conversion of the non-hair cells into hair cell-like cells. So they can activate the expression of those hair cell-like proteins. So then we wanted to know whether or not we could get 801 to work in the mature cochlea. Unfortunately, the expression of 801 all by itself doesn't actually reprogram supporting cells in the mature cochlea. And so we know from various other work that in the neonatal supporting cells, and so these are very early immature cells, the enhancers and promoters that 801 needs to target are actually are accessible in supporting cells. And so we can get the activation of those hair cell proteins. However, as a supporting cell matures over time, these regions called enhancers are no longer accessible to 801, and so we don't get the production of those hair cell proteins, and so we can't convert them into a hair cell-like cell. However, transcription factors often work together. So it's possible if we express multiple transcription factors at the same time, such as GFI1 and POW4F3, which are factors that are very important for the development of hair cells, that we can overcome these genetic barriers and further reprogram these cells into hair cell-like cells. So when we targeted the cells of the mature cochlea, what we found in control is that, again, we have the 1 row of inner hair cells, the 3 rows of outer hair cells, and their various associated supporting cells that we're targeting with these transcription factors. If you express all 3 transcription factors in the ear, we see on this very lateral edge of the organ of Corti, these cells that express this supporting cell tracer in magenta, and they also express the hair cell protein seen in green. And so this indicates that these cells started their life as a supporting cell. After we reprogrammed them, now they have become a hair cell-like cell. And when we can quantify these and see that there's a significant increase in the number of these cells after we have done our manipulation, after we have expressed our 3 transcription factors. And so what this means is that supporting cells require all 3 transcription factors for reprogramming. Something else that we noticed during this project is there's a lot of magenta cells. They all received the expression of all 3 transcription factors. However, only a small number of them are actually converting into a hair cell-like cell. So what could potentially be stopping other supporting cells from being reprogrammed? We know during development and during that period of time when the supporting cells can spontaneously regenerate into a hair cell, that signaling from the hair cells to supporting cells can actually control their identity. And it actually restricts those cells from becoming a hair cell-like cell. And it essentially tells them, hey, you can't become like me, you have to be something different. We don't currently know very much about how this might be working in the adult cochlea. And so we asked whether cochlear hair cells are still controlling cell fate decisions in the mature ear. So to be able to do this, we first needed to ablate or kill all of the hair cells in the cochlea. We did this using a genetic mouse. And after we killed hair cells, we expressed our transcription factors to see whether or not we could get an increase in reprogramming. We can see here in the control tissue that there is a single hair cell remaining from the original hair cells following our hair cell killing manipulation. We can also see that permanent dye that we expressed inside the supporting cells in magenta. So all of the supporting cells are still there. They're just not converting into, and they're not regenerating hair cells. However, if we express all three transcription factors in those magenta cells, in those supporting cells, we can see a significant number of them are now activating hair cell proteins. And we can see that through the co-labeling of a magenta cell and a green cell. And again, when we counted this, we see that there is an increase in the number of these co-labeled cells if you kill the original hair cells first. And so most of these supporting cells actually require hair cell loss to be able to respond to transcription factor reprogramming. And so a quick summary of this particular section is that in control cochlea, where the hair cells are all still there, the genes that 801 and GFI1 and PAL4F3 need to access are inaccessible, or likely inaccessible. However, if you first kill the original hair cells, now these genes seem to become more accessible. And if you express the three transcription factors, now you can convert those sensory or those supporting cells into hair cell-like cells. And so we were very excited by this because what this suggests is that any potential gene therapy that is going to involve these three transcription factors is going to be able to convert the supporting cells around the hair cell lesion preferentially or better potentially than others. And that's still a question that we're working on addressing. So we also wanted to think about all of these other cells in the ear as a potential target for reprogramming. We think that these are an important target because of something called the flat epithelium. And so inside the ear, the sensory cells are epithelial cells. And in a healthy organ of Corti, we can see that there is this dynamic organ where we have the hair cells and supporting cells. However, following severe damage, we lose not only the hair cells, but the supporting cells as well. And so we wanted to know whether or not this flat epithelium is a potential target for a genetic reprogramming. So one leading hypothesis about how the flat epithelium is produced is where cells following this severe damage, the hair cells and supporting cells are lost, and cells of what are called the inner and outer sulcus, which we see here in blue and white, they actually migrate in and form a scar that replaces the organ of Corti. And so if these blue and white cells are able to respond to transcription factor reprogramming, they could potentially be a therapy for people who have lost both hair cells and supporting cells. So we wanted to ask, are the cells in the inner and outer sulcus, are they responsive to transcription factor reprogramming? In, again, the control cochlea, we can see that there are the normal complement of hair cells. And so this actually represents the entire mouse cochlea from the apical turn all the way down into the middle and down into the base. And so we've basically taken that coil and we've flattened it out so we can look at the whole thing all at once. If we express our three reprogramming factors in the mature ear, we can see 2 weeks later the presence of new hair cells in the inner sulcus, which is in here, the outer sulcus out here, as well as something called the spiral limbus, which is kind of medial to that inner sulcus. And so if we give it additional time, we can see that there are more hair cells produced. And 9 weeks after reprogramming, we actually see a significant increase in the number of cells throughout the entire length of the cochlea. And it appears that almost all cells on the floor of the cochlear duct are able to respond to transcription factor reprogramming. And so the conclusions from the work that we have done thus far, essentially the organ of Corti, in the organ of Corti, the supporting cells require all three transcription factors for reprogramming. Hair cell loss actually primes the supporting cells to respond to reprogramming. And in non-sensory cells, the expression of 801, GFI1, and PAL4F3 will actually reprogram cells into hair cell-like cells by activating various hair cell gene networks. And we can add additional cells with time. So a question that I get very often is, can these animals hear? If we're making a whole bunch of hair cells, you'd think that a lot of hair cells equals better hearing. However, the cochlea, sound detection requires a very tight control of the cellular environment. So the way that hearing works, or sound detection works actually, is sound enters our ear canal and it goes through the middle ear and it causes vibrations within the inner ear. These vibrations are picked up by this thing called a basilar membrane, and the hair cells and supporting cells actually sit on top of that basilar membrane. When that vibrates from the sound vibrations, it kind of gets pushed up into the tectorial membrane, and these little hair projections on the top get deflected. This essentially activates those hair cells, allowing them to communicate to the brain, hey, we're detecting sound. I personally like to think of the cochlea like a Rube Goldberg machine. We'll see if this video will play here. So the energy of this ball bearing is being transferred from component to component to component. And to be able to get that energy to kind of go from the very beginning, where we, in the cochlea would have in hearing, we would have the ear canal, all the way through to the brain, all of these various components need to be exactly where they are, in the orientation that they are, in the shape that they are, etc. And so while we've made all of these additional hair cells, that doesn't actually mean that we've recovered sound, hearing detection. So the kind of moving forward, what we need to kind of figure out is, how can we make the hair cells we need to make in the place that we need to make them, so that we can actually control that cellular environment to recover hearing. So some of the ways that my lab is planning on looking at this is by thinking about the cellular identities of these cells in the hair cells and the supporting cells and the non-sensory cells. So we're activating 3 transcription factors that are associated with hair cell identity. But as I mentioned before, these supporting cells and non-sensory cells have their own set of genes that regulate their identity as well. So it's possible that there are some type of cellular breaks preventing these supporting cells from activating hair cell identity. So what I'm really interested in is, if we can identify these breaks and deactivate them, can we get a better conversion of a supporting cell into a hair cell? And then the second kind of direction that the lab is interested in going is, how can we introduce these reprogramming factors in a clinically relevant way? So a lot of the development of AAV-based gene therapies throughout the literature in the last several years has really focused on targeting hair cells and not the supporting cells. So we still need to develop the AAV-based therapies that can actually target the supporting cells. We know that we can make them almost anywhere. So now we can tailor it down, or we're working on figuring out how we can tailor it down so that we can target the cells we want to target, convert those into the right kind of hair cell. And then in combination with that, if we can figure out what the breaks are, we'll be able to develop novel therapeutics in the future to kind of inhibit those breaks while we are trying to reprogram the cells. So both of these are kind of in the initial stages of development here at the University of Pittsburgh. I'm collaborating with another lab here in the PHRC. And we're really interested in kind of answering some of these questions in the future. With that, I'd like to thank my lab. They are a wonderful team here, as well as my funding and my collaborators. As Carrie said, I've been here for a year, and it has been a really wonderful year, and I'm really glad to be able to talk to you guys today.
Thank you, Melissa. Thank you, Peter. Thank everyone. That was a lot of information, good information, and I think it really highlighted the pathway that we see between, what's going on in cells, in genes, and then understanding, really what's on the horizon for care and advancements in care in the future. And we hope and we know that we are going to be on the cutting edge of that here in Pittsburgh. Quick question, because I've gotten this, many of us who work alongside the faculty at the foundation have gotten these questions and both of you are coming relatively recently from different universities, from different health systems. And I want to know why, before we get into all the questions, why did both of you choose to come to Pittsburgh? Maybe I'll start with Melissa and then hand it over to Peter.
Yeah, so I was really excited about really the PHRC. So it's this, group of younger faculty that are really interested in understanding hearing from the cochlea to the cortex. And they're really interested in bridging the clinical and basic science research. And so really it was the team here at the university that really excited me about coming here.
Yeah, I'm going to add, I say this bullishly, I really think that we're going to be the center of hearing in the world, and we have the potential to do that. We're becoming that, and that's what really excited me. It's very rare that you have an excellence in clinical medicine, excellence in science, that you put that together with an excellent university and in an incredible community. I think I really have been impressed by how Pittsburgh proud people are, and the ability to give back and support their community. And I think all of those things rarely exist. I mean, you can get one of those things, but all of those things rarely exist anywhere in the world. And that's when I saw that, when I interviewed you here, I said I had to be here. So, to join people like Melissa and the other scientists here is an amazing opportunity.
Thank you so much. So one question that we get a lot it's from people who are coming in to who are looking for an appointment to be assessed because they've had unexplained hearing loss. And for a patient, anytime you have anything that's unexplained, really you can't assign any particular cause to it, or no one up to this point has been able to assign any particular cause to what's troubling them, like hearing loss. That can be really discouraging, and it can be kind of like a turnoff for people if they're like seeking answers. So Peter, when we talked several months ago, one of the things that you mentioned was about how we're going to kind of eradicate like the idea that so much hearing loss is unexplained. So I wanted to know if you could talk about that a little bit.
Yeah, I mean, the medical term for unexplained is idiopathic, and that's kind of a fancy way of saying it just happens. And I don't think anything in medicine just happens. There's a reason for it. And I think the clinical team here, me itself, are equally as frustrated as the patients are when we bring up the term idiopathic. And so I think one of the goals of the research team here and the clinical team is to get rid of that word idiopathic. We want to know the reason why. And so, we're addressing that on a research level and a clinical research level. So some of the things that we're doing to look at that is, we're redefining how the immune system in the ear interplays with hearing loss. We're trying to look at how some repurposed drugs can be used potentially and look at the traditional idiopathic nature of things. So I think, that's a goal, a long-term vision for our whole group, is to get rid of that word idiopathic. And we're doing that systematically through our clinical practice, bringing some new potential treatments or ways of thinking about patients for that, but also going back to the lab and saying, well, look, can we get biomarkers? Can we work and research these patients that we really don't know what was going on, and not think that's okay, that we really need to go back and think about why that is with our amazing science team.
One other thing as a follow-up to that, you mentioned the importance of time and having kind of a rapid response approach to whenever someone does have sudden hearing loss or some issue with their hearing that's sudden. Can you talk about that at all?
Yeah, that's right. And as Melissa just presented, the ear is one of the rare places in our body that doesn't regenerate. So once it's dead, it doesn't come back. And until Melissa can figure it out, we're not going to have a better solution. So, there's a small window when the injury is potentially reversible. Whatever that has, it hasn't yet finished. And so, we generally think that's probably within the first 6 to 8 weeks of sudden sensorineural hearing loss. So we have a sudden inner ear hearing loss, and of which there are some people that demonstrate reversibility. We are looking at a lot of steroid injections in the ear, steroid tablets. There's some other things that we could potentially do. We're actually trying some new immune modulators in that, but we have to get them in that critical window. If you get to us after 8 weeks, we're having a very different conversation with you than if you get us before that. So it's kind of a good thing. If you notice a sudden hearing loss, it could just be wax, or it could be your inner ear going, and we say just rush and get an audiogram and get it tested, because we really treat you as an inner ear emergency if your inner ear is dropping off function.
I wanted to bring that up because we do have quite an audience here listening. So that's something for people to keep in their back pocket, to tell people that or may encounter that, it's important to be evaluated, soon after any sort of hearing loss, or like Dr. Santa Maria said. So maybe now I'll get into some of the questions that we have from the audience. Speaking, Melissa, about some of your work, and you discussed the genetic reprogramming of certain cells. Does that depend on the type of injury, the type of deafness, if it's congenital, if it's age-related? Does the ability for a cell to be reprogrammed kind of depend on what happened to it?
Absolutely. And so as we think about, this ear in hearing as a bit of a machine, like a Rube Goldberg machine, right? There's a lot of different components. The hair cells are only one component. And so if the injury isn't a loss of hair cells, then the reprogramming that I'm working on isn't really going to be able to help that injury. And so you mentioned the genetic versus acquired hearing loss. And so the idea behind genetic hearing loss often is that a gene is mutated, and so we don't have the right protein to make that cell functional. So sometimes it's still there, sometimes it's not. And so if the cell is still there and that protein is missing, my reprogramming approach isn't really going to be useful for that specific type. However, if that cell is no longer there and we need to make a new one, we could potentially reprogram it and then have another layer of the gene therapy to put that protein back, essentially. So it definitely depends on what the injury is.
Thank you so much. I think I have one more question for Dr. Santa Maria, and then I know he has to run off to a surgery. Is that okay? Do you have time for one more?
Yeah, of course, yeah.
Okay. Here's the question. Oh, I lost it. Sorry. There's a big list. Do you see a risk, or is there a risk of further damaging one's hearing by using over-the-counter hearing aids?
I think there's potential to be damaging to your hearing if you turn anything up too loud. And so I don't think so. Most of the things that are sold over the counter are very safe in that regard. So yes, there is the potential if you just get them and you crank them up, and you do that without anything. But it's very rare that people are going to do that. And I think that they've been designed to be able to be very safe. And that kind of goes on to use of earbuds and personal sound devices. People don't realize that most of them are at damaging levels if you turn them up. If you turn them right up, and young kids are listening to them, especially, very, very loud. And so they are damaging. And so you just have to be very careful. But with the over-counter listening devices and hearing devices, they are very safe as a general statement there. But of course, they can be open to misuse.
Just as a quick follow-up, there are a lot of new over-the-counter hearing aids that use Bluetooth technology, and we have a question from someone in our audience about the safety of using Bluetooth so closely to, your anatomy, if there's any sort of frequency radiation that a user might be concerned about.
Yeah, I mean, it's a good question. At least the research shows that probably not. And I think the biggest research group that we've looked at is what's the worst that a frequency or a radio frequency of Bluetooth could do if it's next to your ear? It's probably giving you a tumor in your ear, right? So, we've looked at very large cohorts. Most of them have come from the Danish cohort, where they've looked at hundreds and thousands of people over decades looking at cell phone use and use of devices around the ear, and they've shown conclusively that there's no evidence that any ear tumors come from using those sort of things so close to your ear. So, of course, that's often a common thing that's kind of touted, but at least the research there, epidemiological research, backs up that you're very likely safe from those things.
Thank you. I know you have to get off to a surgery, so we're going to say goodbye to Dr. Santa Maria, but I know you will be a frequent webinar contributor in the future, so thank you. And then I do have a couple more questions for Melissa, if that's okay.
Thank you.
Okay. Someone asked about like this hair cell regrowth technology that you're working on, this therapy. Would it have an impact on people struggling with tinnitus? Does that affect your hair cells at all?
Potentially. And so it's going to really depend on the cause of the tinnitus. And so that is, again, something that I am not personally an expert in tinnitus. However, there's different things that can go into causing tinnitus, and it's all going to depend on where the injury is. So if the tinnitus is the result of the loss of sensory hair cells, then it could potentially help with the tinnitus.
Thank you. I know we have many people, tinnitus is one of our most popular topics. We have so many people who are struggling with it. It's a terrible thing to have to live with and cope with all the time. So any positive news that we're
Yes.
doing in the department, we like to share. And I'll just put a plug out that we will continue to have future webinars focusing on tinnitus and the advancements in tinnitus treatment and therapies, as well as management strategies. We will continue to have those webinars. We do have another interesting question. Do you think that there could be an artificial replacement for hair cells? Something not within your own body, but something that's implanted? I don't know.
That's essentially what a cochlear implant is to some extent. And so it's always possible. I know there are groups working on a kind of light activated cochlear implant type thing. But otherwise, I'm not really sure if there is a potential to really kind of individually populate small devices inside the ear that would replicate what a hair cell does.
I see. Okay. And then finally, I'll just kind of ask about just like your hopes for the timeline of kind of like, the steps that this work will have to undergo to kind of get into the trial process. Do you have anything you could share with our very interested audience about, people always want to know when they can access something like this?
Yeah, I would say it's going to be a while, unfortunately. So we are still at the state where we're trying to understand the kind of molecular and genetic underpinnings of how to make what the cells that we need to. And again, we have not yet recovered hearing in even mice. And so all of the work that I have done so far has been using models that are not clinically relevant at all. And so transitioning that into a clinical model, understanding the genes we still need to investigate and activate, the genes we need to turn off, how to do it in a way that's going to produce the best potential for making the cells we need them where they need them to be. It's going to be a while. And I'm always very hesitant to give a potential timeline just because there's so many things that can still go wrong. There's so much work left to do.
Well, thank you. And as things happen, updates that come out, the Eye and Ear Foundation works closely with individuals like Dr. McGovern, Dr. Santa Maria. We will make sure everyone knows about new things that are coming out, new trials, new advances. And you can always contact us through our website or our emails. You've obviously found us in some way. So I think with that, thank you for that forthright answer. I think we'll go ahead and end for the day a little early. But thank you very much, Dr. McGovern and Dr. Santa Maria. You are off operating already. We appreciate your time and the presentation that you put together for us today, and we look forward to more. Stay tuned for a recorded version of this posted to our website and emailed. And if there are personal questions that kind of need a follow-up answer, we will get those to the docs and you will get an answer to you mailed directly. So thank you so much, everybody. Enjoy the rest of your afternoon and your weekend.